my presentation will focus on Tay Sachs disease. Tay Sachs disease is a rare inherited condition passed down from parents to children. Tay Sachs disease destroys nerve cells or neurons located in the brain and spinal cord over time. Tay Sachs usually affects infants and the symptoms can appear as early as 6 months old and as late as 3 years old. This deterioration of neurons causes development to slow down. Muscles necessary for movement often become weak, and infants will lose control of motor skills such as crawling and sitting. They also can become sensitive to loud noises. As the disease progresses, children can experience a loss of vision and hearing, trouble thinking or problem solving, and even seizures. Those affected with Tay Sachs disease usually only live into early childhood. The genetic basis of Tay Sachs disease is that it is caused by mutations of a single gene, the hexagene, located on chromosome 15. This gene is necessary for making an enzyme that has a critical role in neurons. This enzyme is created and then transported to lysosomes, a cellular structure responsible for the breakdown of toxic substances in the cell. Normal function of the hexagene creates the enzymes, and the lysosomes are able to recycle and break down toxins and expel them out of the cell. However, mutations associated with Tay Sachs cause the enzymes to not be created and the lysosomes stop working. Then, the toxins will accumulate in the cell, causing damage to the neurons over time. Oftentimes in science, key findings propel our understanding of a disease or a condition. In order to better understand Tay Sachs disease, a team of researchers investigated sections of DNA that were located on either side of the hexagene. The researchers first isolated DNA they were interested in and then sequenced regions of interest. This sequencing showed what nucleotides were where and the order they were in. The researchers then, with these sections of DNA that they were interested in, made lots of copies of these sequences. After they made the copies of these sequences, they ran multiple tests to compare them to DNA from an unaffected person to check for any changes or mutations. These comparisons were done by looking for heteroduplexes or mismatched DNA. The second way that they did these comparisons were looking for single-strand conformational polymorphisms. Using these two methods, 5 new mutations were found. These mutations are separate from the previously understood mutations of the hexagene. These 5 mutations were part of the hexagene and also on either side. These new mutations identified could lead to insights into the causes of Tay Sachs disease. For example, the heteroduplexes found in the hexagene in the figure above can change the expression of the hexagene even if it's not located in the gene itself, because the order of the nucleotides will change. Understanding these mutations and what effects they have can lead to a more complete picture of Tay Sachs disease and how to fix it. However, oftentimes it is important to understand the risks of Tay Sachs disease. Oftentimes, a family will get a sample of their DNA from a skin cell, sequence this DNA, and allow a lab to analyze it and send them the results. This is important to understand the risks of passing Tay Sachs disease down to a family member. As gene therapy and gene editing technology become more common and more effective, it is important to address the benefits it could have for the treatment of Tay Sachs disease. Gene therapy could be used to delete or remove a mutant form of hexagene and replace it with a properly functioning hexagene. This would restore proper function to the lysosomes and allow the cell to destroy the toxins. The continuation of research into the causes and the genetics associated with Tay Sachs disease will hopefully lead to possible treatments and cures.